Hello everyone and welcome to today's live broadcast, Neuroanatomical Substrates Underlying Attentional Processing, presented by Dr. Josh Burk. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots, the leading social networks website and provider of virtual events and webinars advancing scientific collaboration and learning. Here's how the presentation will work today. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see the presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located on the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Josh Burk, Associate Professor and Chair of the Department of Psychology at College of the William and Mary. Professor Burk received a bachelor's degree in psychology from the University of California, Davis, his master's and doctorate in experimental psychology from the University of New Hampshire, and completed a postdoctoral fellowship at the Ohio State University. He joined the William and Mary faculty in 2002, where he is currently an associate professor and chair. Professor Burk was also the director of the neuroscience program in 2011 to 2015 and is currently a faculty affiliate in that program. Professor Burk's laboratory studies the neural basis of attention, learning, memory, and impulsivity. He is largely focused on the role of cholinergic projections to the cortex in the processes and how these projections are regulated by neuropeptides, orexins, or hypocretins. His laboratory has been funded by grants from the NIH, NARSOD, and the Virginia Tobacco Settlement Foundation. His research interests have expanded the study on how neurodiverse students can be more accepted on campuses. He has earned some awards, including the Phi Beta Kappa John D. Rockefeller Award of Advancement of Scholarship and a Plumeria Award for Faculty Excellence. I would now like to turn the presentation over to Dr. Burk.
Great, thank you, Susie, and thank you to LabRoots and the organizers of this event for the opportunity to speak with you today. The title of my talk, as Susie mentioned, is Neuroanatomical Substrates Underlying Attentional Processing. And as such, I wanted to divide this talk up into two different components. During the first part of the talk, I'll discuss some of the brain mechanisms that are involved in attentional processing. And then during the latter part of my talk, I'll discuss a little about how the circuitry becomes altered in neuropsychiatric conditions in which attentional processing is affected. So to begin with, I wanted to describe what's been a large focus of our efforts and other labs as well, trying to understand the role of basal forebrain cholinergic neurons in attentional processing. So what you're looking at here is a sagittal section, a schematic of a sagittal section of a rat brain. And what's being depicted is an area here, the basal forebrain. This is where cell bodies are located that then project to widespread areas of the cortex, and that's being depicted here. So we've studied the role of different aspects of this pathway in a measure of attention. So I want to take a minute to describe this measure of attention because it'll be something that I'll be discussing quite a bit throughout my talk. So the way that the task works is that rats are trained in chambers that contain a central panel light, and that's being depicted, you can see that right here. And there are two levers that are that can be extended and retracted. One of the levers is here, and the other one is right here. So the way that the task works is that on half the trials, the central panel light is illuminated. In most of our experiments, this is for 500, 100, or 25 milliseconds. This can occur randomly throughout the session. If the light's illuminated, then a press on one of the levers is considered correct and rewarded with tap water in most of our experiments. A press on the other lever would be considered incorrect and would be scored a miss. So a correct trial would be scored as a hit, and an incorrect trial would be scored as a miss. On the other half of the trials, there's no signal that's illuminated. So the central panel light is not illuminated. And in these cases, the rules are reversed. So a press on the right lever in this specific example would be considered a correct rejection and rewarded. A press on the left lever would be a false alarm and considered incorrect. And so the animals need to detect whether or not the light was illuminated. Other ways that we make this task attention demanding is that we vary the inter-trial interval so that the animals can't time when specific trials are going to happen. And so we typically, in most of our experiments, this is 9 plus or minus 3 seconds is the inter-trial interval. So just to give you a sense of general performance in this task, along the y-axis is showing percent accuracy either on signal trials in the sense of percent hits, or correct rejections that would be accuracy on the non-signal trials. And so for the hits, what you can see is on the left portion of the figure, it says log signal length along the x-axis, and this is the signal duration. In this particular experiment from Joe McGaughey and Martin Sartor, they varied the signal duration such that they were 550 and 25 milliseconds. And so what you see is signal duration dependent performance with accuracy higher following longer signal durations and accuracy decreasing at shorter signal durations. And then just a general sense of performance on the correct rejections, but because those are no signal trials, the light's not illuminated, there's no signal duration to take into account. One other measure that I'll be mentioning on some of the slides is a vigilance index, and this is a measure of accuracy, sort of overall accuracy on the task that takes into account accuracy on signal and non-signal trials. So one of the experiments I want to describe early on was used an immunotoxin, 192 IgG saccharin. This is a toxin in which there's an antibody, 192 IgG, that's coupled to a ribosome inactivator, saccharin. And so 192 IgG binds to the P75 nerve growth factor receptor, which is located exclusively on cortically projecting cholinergic neurons within the basal forebrain. So this immunotoxin then binds to the P75 nerve growth factor receptor. Saccharin gets taken up via endocytosis into the cell, and then saccharin can serve to inactivate ribosomes, leading to cell death. So this is a way to selectively lesion the cortically projecting cholinergic neurons and then examining effects on attentional performance. In this early work, there were several papers that McGaughey and Sartor published related to this area, so I've just picked one of them here that they published in 1999. Just to give you a general sense of the typical findings. So here, this is a measure of accuracy on the signal trials or on the non-signal trials. And so what you're seeing here is that this is the sham lesion animals are being depicted right here, and then the lesion animals are below them. And what you can see is that the sham lesion animals are performing significantly better than the lesioned animals. And also, when we look at correct rejections, they're showing a little bit of a decrease here. Typically, there's no effect seen on correct rejection. So it's a relatively selective effect on signal duration that gets observed following corticalcholinergic deafferentation. And so this is important because it suggests that the animals were just performing poorly on all trials, perhaps that they just don't know the rules of the task. This suggests that because there's no significant effects on correct rejections, that the animals are still responding based on the rules of the task. They simply are worse on the signal trials. So one of the things that my lab became interested in is trying to understand whether there are specific task demands that might be sensitive to loss of cortical acetylcholine. That is, are there specific aspects of the task that when cholinergic neurons are lesioned that are important for the deficits that McGaughey and Sartor observed? So what we did was we had an experiment in which we had animals that were given corticalcholinergic deafferentation, as I talked about earlier, with 192 IgG saccharin. What's being shown on the left column here is a sham lesion animal from that study, and on the right is a lesioned animal. And so what you can see lower power along the top is a sense that there's darker areas showing some of the acetylcholinesterase fibers have been stained for. Here's an example of what I'm referring to. And in the lesioned animal, so it's a little bit lighter. When we look at higher power under the magnification, either in primary motor cortex or primary somatosensory cortex, what you can see is quite a few fibers in the sham lesion animal, relatively few in the lesion animals. And when we quantified this, we ended up with about a 77% loss on average in the lesion of corticalcholinergic fibers in the lesion animals relative to the sham lesion animals. So then we train the animals in which we manipulate a single aspect of the task at a time. So first we introduced sham lesion and lesion animals to a variable signal duration. We then introduced them to exclusively a short signal duration to examine if these specific aspects of the task were important for revealing deficits of the lesion animals. We also varied the intertrial interval, and we tried decreasing the intertrial interval as well. And so overall, what we observed is when we manipulated any single task parameter, we did not observe deficits or differences in performance between the lesion and the sham lesion animals. However, when we then train the animals on the full attention task with a variable intertrial interval and variable signal durations, so basically the full task that I described earlier rather than just manipulating single parameters, what we observed is a significant decrease in performance in the lesion animals compared to the sham lesion animals. So what this suggests is that it's something about the attentional demands of the entire task and not any specific component of it that's important for revealing deficits of the lesion animals. Other experiments that we've conducted to try and look at the role, and this was done as part of my time when I was in Martin Sarter's lab, was to use microdialysis to be able to sample acetylcholine levels during attentional task performance. I'm sure many of you in the audience are familiar with this. This is a procedure in which artificial cerebrospinal fluid is perfused through an inlet and via semi-permeable membrane. Components like acetylcholine will move from an area of higher concentration to an area of lower concentration if they're small enough to pass through the membrane. This can be collected through an outlet, and then levels of acetylcholine in our case could be quantified using HPLC. And so this is just showing you a sample here of a rat being tested in that attention task chamber that I was showing you earlier, while tethered for microdialysis. And so we also trained rats in some control tasks as well, because initially if we saw elevation in acetylcholine levels during the attentional task performance, well, there's a lot of factors that go into that test. So the animals are getting rewarded, they're pressing levers, so it could just be the motor activity or reward level that's important for elevating acetylcholine levels. So we included control tasks that matched performance in terms of amount of lever pressing the animals did and amount of reward received. And those are described here as a retracting lever task and an FI9 task that we used. And so what you can see here, this is depicting baseline levels right here of acetylcholine in all those three different in rats that were trained in all three of those different types of tasks. No differences in baseline levels. And then when the tasks were initiated, you can see the dark bar along the bottom near the x-axis. We saw a significant elevation in acetylcholine levels, even in our control tasks, which shows that it was important to include these tasks because cortical acetylcholine levels are elevated even by tasks with minimal explicit attentional demands, but that match in terms of amount of lever pressing and reward that the animals receive. However, what we observed during attentional task performance is that there's a significantly greater elevation in cortical acetylcholine levels during when the animals were performing an attentional task relative to the control task. And so overall, what this line of experimentation has showed is that not only does loss of acetylcholine disrupt attentional performance, but if we look in the intact system and measure acetylcholine levels while the animals are performing the attention task, that including explicit attentional demands will produce significantly more elevated levels of cortical acetylcholine compared with matched control tasks. So an additional question that my lab has been asking is to try to figure out the role of cholinergic receptors that might be contributing to performance in these tasks. So essentially asking the question, what are the effects of blocking different types of cholinergic receptors? And so what we initially did was we administered systemically either a nicotinic receptor antagonist mecamylamine or a muscarinic receptor antagonist scopolamine. And what you're seeing here is measures of accuracy relative hits following nicotinic receptor blockade. And on the left panel, we did not observe any significant effects of blocking the nicotinic receptors on attentional task performance. It's possible that we didn't use sufficiently high doses of mecamylamine to observe effects. So in the figure on the right, what I'm showing you are the number of omissions per session and across dose along the x-axis. And what you can see is that the highest dose did significantly elevate omissions, which suggests that we were at a sufficiently high dose. We just didn't observe differences in accuracy when the nicotinic receptors were blocked. We looked at the effects of blocking the muscarinic receptors with scopolamine, relative hits along the y-axis here, signal duration along the x-axis. What you can see is that there's a dose-dependent decrease in accuracy following muscarinic receptor blockade. And so this led us sort of on a path to focus on the muscarinic receptors more specifically. We reasoned based upon the literature that because the M1 receptors are largely located postsynaptically, that blocking those receptors may be responsible for some of the deficits that we had observed with scopolamine. So we used dicyclomine at different doses, you can see along the x-axis, relative hits along the y-axis. And what we observed is that higher doses, that blocking the M1 receptor with dicyclomine using systemic administration impaired performance. We went on to also distract the animals by flashing a house light in the back of the chamber throughout the testing session. And what we observed, which was interesting, is that even at lower doses than we had observed deficits previously, we see declines in performance. So essentially, by increasing the attentional demands, by distracting the animals, we shifted the dose response curve so that the animals were now showing deficits in performance at lower doses compared with what we had seen in our standard attention tasks in which we're not distracting the animals with the flashing house light. We wanted to know whether these effects were centrally mediated because these were IP injections that we were doing. And so what we did was into the lateral ventricle, we administered pirenzepine, an M1 receptor antagonist, and we did see all those not huge effects. They were statistically reliable. There's a significant decrease in detecting the longer signal durations when pirenzepine was administered compared with vehicle administration. As I'm sure all of you in the audience know, these muscarinic receptors are metabotropic receptors, which means they're coupled to G proteins. In the case of the M1 receptor, one of the things that's activated following stimulation of this receptor by acetylcholine is an elevation of protein kinase C. And so one of the questions we then wanted to ask is what are the effects of inhibiting protein kinase C on attentional task performance? So again, along the y-axis, it's showing accuracy on signal trials, percent hits, log signal duration along the x-axis. And so what you can see is at the highest dose when we administered calorethrin chloride, a PKC inhibitor, that we decrease performance in our attentional task. And because PKC is located sort of in widespread areas, we wanted to see whether this effect was selective. So we also looked at accuracy on the non-signal trials, percent correct rejections, and we observed no effects of PKC inhibition on non-signal trials. So this suggests, much like what was seen earlier in the earlier work with selective lesions of corticocolinergic neurons, what we observed is that there's a selective decrease with PKC inhibition on signal trials with no effect on the non-signal trials. We then wanted to look at the flip side of this because we basically mapped out how we could impair the muscarinic receptor system followed by the M1 receptor system and then PKC. So we wanted to flip that around and see in lesion animals, could we reverse attentional deficits when we administered an activator, a PKC activator, bryostatin 1. And so what we observe, and I'll point out the difference from vehicle here where the lesion animals are performing worse than the sham lesion. When we administer a low dose of bryostatin 1, we observe a significant increase in performance in the lesion animals. So we're able to see improvements in task performance of the lesion animals following PKC activation. So we previously observed PKC inhibition disrupted performance. Here, PKC activation can enhance performance of lesion animals. We also saw improved performance of the sham lesion animals at higher doses. We're still puzzling a little bit about the nature of the dose response curve, but we are encouraged by the general pattern of results but the animals seem to be able to show improvements in performance following PKC activation. We've also looked at an M1 allosteric agonist. I'm not going to show you those data because we didn't find any significant effects when we used an M1 allosteric receptor agonist. Where we seem to see the strongest beneficial effects are when we activate PKC. We also wanted to look at another aspect of research in my lab that we're interested in is looking at regulation of these basal forebrain cholinergic neurons. And so the pathway that's being highlighted right here originates within the hypothalamus and projects to the basal forebrain. The system that we focused in on releases neuropeptides called orexins or hypocretins. And these hypocretins or orexins have been shown to, when infused into the basal forebrain, to elevate cortical acetylcholine release. So we were interested in whether the system known to modulate basal forebrain cholinergic neurons would affect attentional performance. And so some of our earlier work, we administered drug SB to be able to impair orexin 1 or block orexin 1 receptors. And what we observed is a decrease in accuracy when we administered this drug. And you can see the different doses that were used in the legend here. And when we looked at overall performance, the VI measure along the top, we saw a significant decrease in performance. And when we looked at accuracy on signal versus non-signal trials, I'm showing you the signal trial accuracy here, we observed a significant decrease in performance on the signal trials. We also observed, and I should have mentioned on that previous slide, that was systemic administration of the antagonist. We've also looked at infusions directly into the basal forebrain of orexin 1 receptor blockade, and again, showing you VI in the figure along the top and relative hits along the bottom, the figure along the bottom. And we observed a significant decrease in performance when we administered an orexin 1 receptor antagonist into the basal forebrain. So that suggests that we were able to localize the effects to the basal forebrain. We've also looked at the effects of orexin A on attentional performance. So much like we had looked at PKC inhibition and then PKC activation, we've also looked at the effects of blocking orexin 1 receptors and then stimulation of orexin receptors with orexin A. And so along the top here, this is a measure in which we had the session set up so that there was an initial block of 54 trials in which the animals got our standard attention task, followed by a block of 54 trials in which we presented that distractor where we flashed the house light in the back of the chamber. And essentially what we're looking at are different scores in terms of performance during the distractor compared minus performance during the initial block of the standard attention test. And so what we observe here, this is just to reference the measure here. So the distractor is producing greater decreases in accuracy when measures are over in this part, so when they're above 0. So measures when it's a negative value, that suggests that they're doing better during the distractor session. And so initially what we observed, these are in intact animals in which we administered orexin A into the lateral ventricle. And what we observe is that orexin A is able to reverse deficits due to the distractor in attentional task performance. That's being highlighted by the asterisk and the line comparisons here. And we see this in our overall measure, our vigilance index, or also referred to as the SAT. This measures sustained attention. And along the bottom here, I'm showing you accuracy on signal trials selectively relative hits. And again, just like we've seen in many of our other studies, this effect was largely due to changes in accuracy on the signal trials. So orexin A is able to, when administered into the lateral ventricle, is able to reverse deficits due to the distractor. Finally, we also wanted to look in lesioned animals whether we could reverse deficits due to the distractor. So this is a graph showing you accuracy on the shortest signal trials here. And here when we administered orexin A, what we observed, again, is initially during vehicle performance, the lesion animals are more affected by the distractor, performing worse compared to the sham lesion animals. However, when we administered orexin A, the lesion animals benefited more and were now showing less of an impairment during the distractor session. So overall, these results do suggest that orexin A may be beneficial in reversing attentional deficits associated with dysfunctional cholinergic corticopedal system. So I want to move to the second part of my talk, since I've described a lot of the neural mechanisms that we've focused on, looking at the role of orexins and different receptors within the cholinergic system, to look at during different studies related to different neuropsychiatric conditions in which attention is disrupted, what are some of the studies that we've conducted and results that we found? Well, one of the things that we've looked at is the role of producing smaller lesions to the corticalcholinergic system early in life in adulthood, and assessed whether, as the animals aged, their performance would decline. And this is related to ideas related to Alzheimer's disease, in which there's thought to be pathology or brain difference that's occurring early on in life that becomes exacerbated by the aging process. So what we wanted to test is whether partial damage to the corticalcholinergic system would be sufficient to lead to attentional deficits as the animals aged. So in terms of general experimental design, the animals were trained in that sustained attention task that I was describing earlier until age 16 months. They were then administered 192 IgG saccharin or saline and then tested for the remainder of their lives in that same measure of attention. And so what we expected is that because we were producing relatively small lesions using a relatively low dose of the immunotoxin, we expected that we would end up with no deficits in attentional performance early on, right after the animals recover. But what we expected is that the aged animals would show impairments later in life. And so this is just giving you a sense for the scope or the nature of the lesions. So the left column is depicting at different magnifications a sham lesion animal, and then on the right column a lesion animal. And what you notice is that it's a fairly dark in terms of the sham lesion animals, but it's not completely bleached out in the column on the right. So there are still some acetylcholinesterase positive fibers in the column on the right. When we went to quantify this, looking across different cortical areas, what we saw was that our lesions had produced a loss of about 40 to 60% of corticalcholinergic inputs, varying again a little bit across different brain regions. Of course, we conduct these types of studies. One of the concerns is that perhaps the lesions have just made the animal sick and that there's no selective deficit in terms of attentional performance, but rather there are issues just with general health. One way that we assessed this was by measuring the animal's body weight throughout their lifespan, and what you can see here is that there's no significant difference in body weight between the sham and the lesion animals. In terms of attentional performance along the y-axis, showing you that overall measure of attentional performance, vigilance index and signal duration along the x-axis. When we tested the animals immediately after surgery, there was no significant difference between the sham and lesion animals in terms of their level of accuracy. When we continued to train the animals out to age 21 months and 26 months of age, again, we did not observe any significant decrease in accuracy in the lesion animals relative to the sham lesion animals. Although you'll note at 26 months of age, we're starting to see a slight dissociation between the two groups. And we continued to train the animals to 31 months of age. What we observed then, VI along the y-axis here, signal duration along the x-axis. At 31 months of age, the lesion animals showed an impairment relative to the sham lesion animals in accuracy on that measure of attention. And so when we looked across blocks of trials, so what block represents along the x-axis during block 1, that's trials 1 to 54. During block 2, trials 55 to 108. And during block 3, that's the last third of the trials, 109 through trial 162. And so what we observed is evidence of a sustained attention deficit in the lesion animals when they were 31 months of age, such that they were significantly worse during the third block of trials within a testing session. So basically, as the session continued, the sham lesion animals show no difference in performance. However, the lesion animals show a significant decline as the testing session went on. Another approach that we've taken is to try to understand whether models of schizophrenia have effects on attentional performance as well. And so we've looked at sort of the two classic types of approaches, looking at a hyperactive dopamine system and an underactive NMDA receptor system. And so what we did was, in one study, we looked at, this is percent false alarm. So this is accuracy. For the most part, I've been talking about accuracy on the signal trials. Here in these models of schizophrenia, we're looking at accuracy on non-signal trials. And in fact, what we see when we expose the animals to amphetamine repeatedly, that they make more errors on the non-signal trials. We did not observe any difference in accuracy on the signal trials in animals that have been repeatedly exposed to amphetamine compared to those that had been repeatedly administered saline. Looking at the other component, if we block NMDA receptors, what we observe is the figure on the left here is showing the accuracy on the signal trials, and there's no significant difference between saline-exposed animals and those that were given the NMDA receptor antagonist MK801. However, in the figure on the right, depicting false alarms, when we look at administration of MK801, there's a significant increase in the number of false alarms when MK801 is administered relative to saline administration. And so one of the things that we were interested is in using an allosteric agonist at the NMDA receptor D-cycloserine to try to have beneficial effects in terms of reversing deficits following MK801 administration. Here what I'm showing you is administration of entirely vehicle. I should mention this is percent false alarms along the y-axis. So you can see what the false alarm rate was following saline administration, our vehicle group. In the animals that received MK801, they showed a significant increase in the false alarms as we had seen previously. In the groups that were given MK801 and D-cycloserine, we observed a reversal, a dose-dependent reversal in the attentional deficits following MK801 administration. So this suggests that there's potential for reversing these types of deficits by acting at the NMDA receptor. Further experiments in our lab are looking at whether perhaps orexins are targeting the cholinergic system in other ways could reverse some of these deficits as well. So some take-home messages from my talk today. Acetylcholine, this hopefully I've convinced you is critical for normal signal detection and this attention-demanding task. And we look at the different receptor mechanisms involved. The muscarinic receptors appear to be critically involved in mediating the effects of acetylcholine on signal detection. On non-signal trials, this appears to be more sensitive to manipulations that mimic some of the neurotransmitter changes in schizophrenia. And so we're, again, doing further work to try to assess how we might be able to have beneficial effects to reverse some of the attentional deficits in those models of schizophrenia. And finally, our work suggests that we may be able to target protein kinase C or the orexin system in different ways to be able to have beneficial effects in reversing attentional deficits following various manipulations. I want to thank the students in my lab who conducted this work. I've had some very hardworking undergraduate honor students who have contributed to these projects. And on the bottom here are the graduate students are highlighted in bold who are responsible for the work that I talked about today. I'd like to thank my collaborators on these different projects, Moore Arnold, Martin Sarter, and especially Jim Fadal's been a longtime collaborator on the orexin work. And I'd like to thank the funding sources as well. And now I'd be happy to take any questions that you have.
Thank you so much for your presentation, Dr. Burk. We want to get right to everyone's questions and input, so here's a reminder how to let us know what you would like to ask. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your questions due to time constraints, Dr. Burk will answer them by submitting the questions via email. So here is our first question. Dr. Burk, please describe expected future directions for your research.
Yeah, So I think a large part of our work is better understanding the role of the orexin system in attentional performance. I highlighted the work that we've done looking at orexin 1 receptors and orexin A. We haven't yet looked at orexin 2 receptors or orexin B in attentional performance. We're conducting those experiments now, and so I think that's going to be an important area for future investigation. In addition, we've been focusing on the orexin inputs to the basal forebrain, but we haven't yet studied, and nobody's well characterized, the attentional effects of orexinergic inputs to, for example, the prefrontal cortex. So I think better characterizing those pathways will be important. In addition, I would say better understanding the role of second messenger systems, like given our data with respect to protein kinase C, is important as well. And so that's another area for future investigation. And then trying to understand whether these can be targets potentially for conditions such as Alzheimer's disease and schizophrenia.
Thank you so much. Another audience member would like to know, could you discuss the role of nicotinic cholinergic receptors in attention, please?
Sure. So one of the things that surprised me when we did some of the early work was when we blocked nicotinic receptors, we did not observe substantial effects on attentional task performance. And so nicotine is well understood to be able to enhance attention. So I think of it as the muscarinic receptor system as being the critical player for attention, and then the nicotinic receptor system may be a way to fine-tune attentional processing as well. So I think of the two systems as working together, with the nicotinic receptor system maybe not being absolutely necessary, but being a way to fine-tune functions needed via the muscarinic receptor system. In addition, one of the ways that this connects with another aspect of my talk is that one of the things that patients with schizophrenia all regularly do is smoke, and it's thought that they may be self-medicating for cognitive deficits through nicotine administration. So one of the things that we're interested in as well is whether nicotine, which stimulates the orexin system, whether the orexin system could be a target for some of the cognitive deficits in schizophrenia.
Thank you, Dr. Burk. We have time for one more question. An audience member would like to know, what are some potential side effects of activating the orexin system in an effort to improve conditions associated with the attentional deficits?
Sure. So I think some of the potential side effects of activating the orexin system are disruption of sleep and increases in anxiety. So for example, the orexin system, when it's activated, would be expected to produce an organism that's more alert. And if this is taken, for example, right before going to sleep, it could certainly cause sleep difficulties. So I think some of the challenges will be impact on sleep, higher levels of orexins if the administration is given at a dose that's too high for a particular individual, then what could happen is that you have increases in anxiety as well. And so I think those are going to be some of the side effects that need to be worked out. And some of the ways to do that might be through better drugs that are better able to target allosteric sites on the orexin receptor. So this is another area of future directions. And there's been very little research and drug development in this area, but I think that could have a huge potential in terms of having having beneficial effects while minimizing the side effects.
I would like to thank you again, Dr. Burk, for your presentation. Do you have any final comments for the audience before we?
Well, I just want to thank LabRoots again for the opportunity to talk with you about this work. And if anybody would like to contact me to hear more, because I certainly didn't describe all the details in the projects, you have a lot of the papers were shown up there. But if you have any additional questions about the nature of our research, I'd be happy to talk further.
Thank you again, Dr. Burk. I would like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available on demand viewing through June 16th, 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward the announcements to your colleagues and that didn't get a chance to be today at today's live event. Thank you again for joining us, and we hope to see you next time. Bye-bye.